Imagine someone hands you a treasure map. Right, an X marks the exact spot.
Sounds pretty good so far.
Yeah, you'd think. So you hike all the way out there, you dig down, you pop open this chest expecting gold, but instead you just find this bizarre, super complex tangle of electrical wires.
And you have absolutely no idea how to read them.
Exactly. There's no instruction manual, no labels, just this massive web of connections disappearing into the dirt. And, that is the exact frustrating reality that geneticists have been facing for, the last 15 years.
It really is.
Because we have mapped the human genome. And through tools like genome-wide association studies, which we usually just call GWAS, we actually know the precise genetic zip codes that are linked to hundreds of complex diseases.
If you have a specific single-letter typo in your DNA, your risk for a certain disease spikes.
But here is the massive catch for you listening. The vast majority of these disease-triggering variants, they don't actually sit inside the genes that code for proteins.
No, they don't.
They sit in the non-coding regions of your DNA, the genetic dark matter. So we know exactly where the disease trigger is located, but we are completely blind to what it physically does to the cell.
It is essentially a molecular crime scene. We have the suspect's home address, but zero understanding of their actual method of operation.
Yeah, a total mystery.
Because a variant in a non-coding region doesn't change the actual blueprint of the protein itself. So for a really long time, researchers were just staring at these statistical peaks on a graph. They knew a disease originated at a specific coordinate, but they lacked the biological machinery to explain the mechanism.
And that brings us to today's deep dive. We are jumping into a landmark science paper by Peter Orchard and his colleagues from the TOPMed program.
Which stands for Transomics for Precision Medicine.
Right. And our mission today is to explore how this team used a colossal data set. We're talking over 14,000 RNA sequencing samples to decode that genetic dark matter.
What's fascinating here is that they aren't just reading the static genome anymore.
They're watching it work.
Exactly. They are using transcriptomics to watch the DNA actually operate in living tissue. They are translating those mysterious coordinates into concrete physical mechanisms using these tools called eQTLs and sQTLs.
Okay, let's unpack those terms, because before we dive into the sheer scale of this study, we really need to understand the basic tools they used.
Right. To truly understand how revolutionary this TOPMed data is, we have to establish the physical mechanism of an eQTL first.
And that stands for expression quantitative trait locus, right?
Yes. So when we say a non-coding variant acts as a genetic switch, we're talking about physical binding sites on the DNA strand.
Okay, picture the DNA strand.
Right. And non-coding DNA is littered with these regulatory regions called promoters and enhancers. And transcription factors, which are just specialized proteins, they have to physically latch onto these regulatory regions.
Like a docking station.
Exactly. They dock there to initiate the reading of a nearby gene by RNA polymerase.
So let me try an analogy. If the transcription factor is a key, the non-coding regulatory region is the lock.
I like that, yeah. And a genetic variant, a single nucleotide polymorphism, or SMP, is basically a tiny mutation that slightly alters the shape of that lock.
Oh, so the key doesn't fit right anymore.
Right. If the lock changes shape, the transcription factor can't bind as effectively, so the RNA polymerase machinery might stall out, or, perhaps it binds too aggressively instead.
Wow, great.
Consequently, the cellular machinery changes the expression level of that target gene. It dictates whether your body manufactures a massive flood of a specific protein or just starves the cell of it entirely.
Just from that one tiny shape change in the lock.
Exactly. That physical disruption of the binding site, that change in expression, that is an eQTL.
Got it. And then we have sQTLs, where the S stands for splicing.
Yes, this operates a bit further down on the manufacturing assembly line itself.
Right, so when a gene is transcribed into RNA, it contains coding regions, which are called exons, and then this non-coding filler called introns.
And the cell's spliceosome acts like a pair of molecular scissors, right? It cuts out the filler and pastes the exons together.
It's like editing a movie. You shoot a bunch of footage, and then the editor chops out the bad takes and splices the good scenes together to make the final film.
That's a perfect way to look at it. And a genetic variant in the non-coding region can actually alter the specific sequence that tells the spliceosome where to make those cuts.
So the editor gets bad instructions.
Right. If the spliceosome gets confused by a variant, it might just skip an exon entirely or accidentally leave an intron in there.
Oh, wow. So you end up with a structurally different version of the protein.
Exactly. You get an isoform. The blueprint of the gene itself wasn't mutated, but the non-coding variant corrupted the cutting and pasting process.
So both eQTLs and sQTLs give us a functional mechanical explanation for these variants that, in the past, would have just looked like weird statistical noise on a GWAS graph.
Precisely. And to map all of these effects, the TOPMed researchers utilized a really clever mathematical framework called colocalization.
Let's break that down for the listener. They took 164 different human traits from the UK Biobank GWS data, right?
Yeah, everything from red blood cell counts to super complex autoimmune diseases.
And they basically overlaid those disease maps with their own transcriptomic eQTL and sQTL data.
Right. Colocalization relies on Bayesian statistics. It's a way to determine the probability that two different signals share the exact same causal genetic variant.
So it's not just, hey, these two things are in the same general neighborhood on chromosome 4.
No, not at all. We are mathematically proving that the specific nucleotide mutation driving the disease risk is the exact same mutation that is physically disrupting the transcription factor binding site.
That lock and key we talked about.
Exactly. When those signals colocalize perfectly, the dark matter is finally illuminated. We suddenly have a causal biological pathway.
Okay, let's do a quick recap of this first major concept for you listening, just to lock it in. We've established that genetic dark matter hides disease triggers. But we can eliminate them by watching how non-coding variants physically disrupt transcription factors. Those are the eQTLs or spliceosomes, the sQTLs.
Right. And colocalization is the mathematical tool that lets us map complex disease traits directly to those microscopic disruptions.
Exactly. Which brings up a huge question to frame our next step. If one non-coding variant can slightly alter a binding site, how many different independent variants could potentially manipulate that exact same gene?
Well, that question leads directly to the core breakthrough of this specific science paper, and it hinges entirely on statistical power.
Because we've understood the basic mechanics of eQTLs for years now. This isn't a new concept.
No, it's not. But transcriptomics is incredibly noisy data. RNA degrades rapidly, and gene expression fluctuates based on all sorts of things, the time of day, what you ate for breakfast, or just minor cellular stress.
It's just a really loud biological environment.
Extremely loud. So to cut through that environmental noise and see true genetic effects, you need an enormous sample size. And TOPMed analyzed over 6,400 whole blood samples, plus another 1,200 lung samples, all uniformly processed.
Which is just staggering scale. And that sheer volume of data is what allowed the researchers to move beyond just the primary signal.
Yes. In the past, geneticists would find the loudest, most obvious genetic switch for a gene, declare victory, and move on.
Because the smaller sample sizes meant everything else was just a blur of statistical static anyway, right?
Exactly. But with 14,000 samples, TOPMed utilized fine mapping algorithms to actually deconstruct that static.
Let's talk about fine mapping because it connects to this really tricky concept called linkage disequilibrium.
Right. So DNA is inherited in chunks, meaning a causal genetic variant is usually inherited alongside dozens of innocent bystander variants that just happen to be located nearby.
They're just along for the ride.
Yeah, and in small studies, they all look like one giant blended signal. You can't tell who's actually driving the car.
But TOPMed's massive sample size let them break those chunks apart.
Exactly. The fine mapping algorithms broke those inherited chunks apart mathematically, revealing that genes don't just have one primary regulatory switch.
They found tens of thousands of what they call secondary signals.
Right. A single gene might actually be governed by 2, 3, or 4 completely independent regulatory switches scattered all across its non-coding region.
Okay, but let me play devil's advocate here for a second. Does discovering a secondary switch actually change the clinical reality of how we treat a disease? I mean, if a gene's expression is broken, does a doctor really care if it's broken by a primary switch or a secondary one? The downstream symptom is the same.
It absolutely matters. It fundamentally changes clinical targeting because it dictates our understanding of disease heritability. Listen to this stat from the paper. The researchers successfully linked over 10,000 GWAS disease signals to specific genes using colocalization.
Okay, 10,000 known disease triggers.
Right. But out of those 10,000, an astonishing 7,000 colocalized with at least one secondary signal.
Wait, really? So for nearly 67% of these disease traits, the actual culprit driving the risk was not the primary genetic switch.
Correct. The majority of the disease signals were operating through secondary or tertiary switches.
Oh, wow.
Think about what that means for drug development. If pharmaceutical companies are designing targeted gene therapies based only on the primary regulatory pathways because that's all the older databases could see, those therapies might completely fail for a patient whose disease is actually driven by a secondary switch.
Because they're trying to fix a switch that isn't even the problem for that specific patient.
Exactly. Without the massive sample sizes of TOPMed, our genetic risk models are effectively blind to the actual mechanisms driving complex diseases.
That makes total sense. And the paper actually provides some highly specific biological case studies to prove this, which I want to get into, particularly with the HK1 gene.
Yes, hexokinase 1.
Right, which is an enzyme that dictates the very first step of glycolysis, basically how cells break down sugar for energy. And it is absolutely vital for energy metabolism in red blood cells.
Right. If HK1 is misregulated, it can lead to severe hemolytic anemia, where your red blood cells are destroyed faster than they can even be made.
And through this fine mapping technique, the researchers isolated 3 completely separate, independent genetic switches for the HK1 gene.
Right, and when they colocalized that data, those 3 distinct regulatory switches perfectly matched 3 different GWAS signals for red blood cell volume.
So the disease trait wasn't just an isolated malfunction, it was the result of multiple parallel vulnerabilities in the noncoding DNA.
Exactly. And they found something similar with the CEBP gene, too.
Oh yeah, which is a critical transcription factor for the innate immune system. It drives the development of monocytes and macrophages. Those are the white blood cells that act like garbage disposals, swallowing cellular debris and pathogens.
And TOPMed identified 4 separate secondary signals for CEBP. And they mapped perfectly to 4 different GWAS signals for monocyte counts in the blood.
4 completely different paths to the same immune response malfunction.
Yes. And if TOPMed were the size of older legacy studies, those multiple independent pathways would remain entirely mathematically invisible.
Okay, let's lock in another quick summary here. By dramatically increasing sample size, TOPMed proved that genes are rarely controlled by just one single primary switch. Almost 70% of mapped disease signals operate through these hidden secondary or tertiary regulatory pathways.
Which absolutely forces us to rethink how we design targeted gene therapies.
Exactly. So consider this scenario as we transition to the next huge finding. If essential genes like HK1 are governed by multiple independent switches, what happens to our understanding of human biology when the architecture of those switches varies wildly across different global populations?
Well, what's fascinating here is that exact scenario highlights the diversity dividend, which honestly is arguably the most critical clinical contribution of the entire TOPMed dataset.
Yeah, scale isn't just about how many samples you have, it's about who is actually in those samples.
Right. And to understand why this matters so much, we have to look at the historical benchmark, which was the Genotype Tissue Expression Project, or GTX.
Right. For years, GTX was the gold standard for eQTL databases.
But GTX, like so many historical repositories, was overwhelmingly Eurocentric. Up to 85% of the samples came from individuals of European descent.
And TOPMed actively dismantled that paradigm, right? They incorporated cohorts with deep African, Amerindian, and other ancestries.
And they didn't just add numbers for the sake of diversity. They added crucial evolutionary depth.
Let's explain why that matters physically. It goes back to that linkage disequilibrium thing we mentioned earlier, the chunks of inherited DNA.
Yes. African genomes, for example, have significantly less linkage disequilibrium, meaning their inherited chunks of DNA are much smaller.
Because human populations originated in Africa, right?
Exactly. Giving those genomes significantly more time to recombine and break apart over hundreds of thousands of generations.
Like shuffling a deck of cards over and over again for a much longer time.
Exactly. And the quantifiable results of testing those very populations in TOPMed are staggering. The researchers uncovered over 11,000 whole blood cis eQTLs, so local genetic switches that are driven by variants that are incredibly rare in populations of European descent.
But highly common in individuals with African or Amerindian ancestry.
Right.
Think about that. Over 11,000 regulatory switches governing basic human biology were essentially unknown to science before this. It's like trying to understand global human language by only reading a dictionary of English slang. Now we finally have a multilingual dictionary.
I love that analogy because when the researchers cross-referenced these diverse signals against older Eurocentric databases to see how many had actually been recorded before, the answer was just 1.3%.
1.3%. That is wild. And this is exactly why polygenic risk scores have historically failed in clinical settings for non-European patients.
Is the exact mechanical reason, yes.
Let's trace the mechanics of that failure for the listener. Because a polygenic risk score is basically just an algorithm. A doctor takes a patient's DNA, and the algorithm sums up thousands of tiny genetic variants to calculate their lifetime risk for a complex disease, like coronary artery disease or type 2 diabetes.
Right, but that algorithm calculates risk by looking at the specific variants, the switches that were identified in its training data.
And if your risk score is trained exclusively on European linkage disequilibrium blocks,
then it assumes a specific, rigid set of primary and secondary switches drive the disease. If you apply that exact same algorithm to a patient of Amerindian descent, whose fundamental regulatory architecture utilizes completely different switches that were invisible in the training data, the prediction model just collapses.
it spits out a vastly inaccurate risk profile, which could lead to terrible clinical decisions.
Yes. The push for diverse genetic datasets isn't just about representation for the sake of optics. It is a strict mathematical requirement for precision medicine to function safely for everyone.
If we don't map the alternative evolutionary pathways regulating these essential genes, our clinical algorithms are just functionally useless for the global majority.
Exactly. It refines our entire map of human biology. We can't claim to understand the human genome if we are only reading 1.3% of the regulatory dictionary for large swaths of the global population.
Okay, let's summarize this diversity dividend real quick. Legacy genetic databases were mathematically blind to over 11,000 local genetic switches because they relied too heavily on Eurocentric samples. And this massive blind spot mechanically breaks polygenic risk scores for diverse populations.
Making diverse data sets a strict clinical necessity moving forward.
Absolutely. Okay, now everything we have discussed so far involves local switches, what the paper calls cis-eQTLs. The regulatory switch sits physically adjacent to the gene it controls on the DNA strand.
Right, the lock is right next to the door.
Exactly. But human biology is an interconnected network. So what happens when a variant on one chromosome physically exerts control over a gene located on a completely different chromosome?
Well, now we are moving into the territory of trans-eQTLs, with trans meaning across.
And these are famously hard to find, right?
Finding a trans-eQTL is an absolute statistical nightmare. With a local cis-eQTL, your search space is limited. You only test the genetic variants immediately surrounding the target gene.
Great, you're only looking in that one neighborhood.
But with a trans-eQTL, you're looking for effects across the entire genome. You have to test every single genetic variant, millions of them, against the expression levels of every single gene, tens of thousands of them.
So the multiple testing burden is just astronomical. You're running literally billions of statistical tests.
Right, and every time you run a test, the chance of a false positive increases just by random chance. So you have to apply a brutal statistical penalty, like a Bonferroni correction, to the p-value just to prove the signal is genuinely real.
This statistical static must be deafening.
It is. You need a colossal data set like TOPMed simply to achieve the statistical power required to hear the true signal over the noise.
And they did hear it. The study identified over 1,700 trans-E genes in whole blood.
Yes. These are genes governed by master switches located on completely different chromosomes.
Okay, let's trace the physical mechanism of how this actually happens, because it's not magic, it's not telepathy. There is a physical bridge between the chromosomes.
Right.
The paper details a fascinating example involving a specific genetic typo of variant called RS169474425. Let's walk the listener through this. This non-coding variant acts as a local cis switch for a nearby gene called ERN1.
Exactly. The non-coding variant disrupts the binding site, which alters how much ERN1 protein the cell actually manufactures locally.
But ERN1 is not just a passive structural protein. It acts as a master cellular sensor and regulator. Specifically, it manages the cellular ER stress response.
Right, and the ER, or endoplasmic reticulum, is basically the cell's protein folding factory.
So when a cell is under stress, the proteins it's trying to make can fold incorrectly.
And misfolded proteins are highly toxic. If the cell doesn't immediately halt production and clear them out, the cell will undergo apoptosis. It will essentially self-destruct to save the surrounding tissue.
And the ERN1 protein is the master regulator that detects that stress and sounds the alarm.
Yes. So because the local non-coding variant changed the baseline production of ERN1, the researchers observed a massive cascade effect. The altered amount of ERN1 protein physically travels across the cell nucleus, binds to different chromosomes, and subsequently alters the transcription of 13 other distant genes.
13 distant genes changed by one local variant?
Right. And one of those downstream targets is XBP1, which is a famous gene that dictates the unfolded protein response to try and save the cell from that self-destruction.
It is quite literally a massive biological domino effect. The non-coding variant knocks over the first local domino by altering ERN1 expression.
Yeah.
That protein physically travels across the nucleus and acts as a transcription factor itself, knocking over 13 other dominoes across the entire genome.
And what's crucial is that a significant portion of these distant trans effects directly colocalized with Nieko's disease signals, particularly for complex blood traits.
Which completely reshapes how we view therapeutic intervention, doesn't it?
Absolutely. Because if a patient presents with a disease caused by the misregulation of 13 different downstream genes, trying to develop 13 different drugs to fix all of them is nearly impossible.
It'd be a nightmare.
But if we map the trans eQTL cascade and trace it all the way back to a single upstream master switch, like that variant controlling ERN1, we suddenly have a highly targeted point of intervention.
You stop the cascade before it even starts. You just grab the first domino.
Exactly.
Okay, time for another recap. Trans eQTLs prove that the genome operates as an interconnected web. A single local mutation can alter a master regulator protein, which then physically travels across the cell nucleus to regulate dozens of distant genes on completely different chromosomes.
Creating massive biological cascades tied to complex diseases.
Right. But this brings up an intriguing logistical puzzle. TOPMed is incredibly powerful at mapping these intricate networks in blood and lung tissue.
Yes, those were the primary tissues studied.
But how does this immense scale compare to what we already know about the rest of the human body? If TOPMed is this massive and powerful, does it make all previous databases like GTEx completely obsolete?
Well, if we connect this to the bigger picture, it brings us to a fundamental trade-off in genomic research: sample size versus tissue breadth.
Okay, lay it out for us.
When researchers compare the new TOPMed data with the older GTEx ectus, it becomes very clear that one does not render the other obsolete at all. TOPMed has this unprecedented sample size of over 14,000, but the vast majority of that data is concentrated in just two highly accessible tissues: whole blood and lung tissue.
Because it's a lot easier to get a blood draw from 10,000 people than a brain biopsy.
Exactly. GTEx, conversely, has much smaller sample cohorts. Sometimes they only have a few hundred individuals for a tissue, but GTEx covers 49 different tissues across the human body.
Right. They have samples from various regions of the brain: the liver, the heart, the pancreas.
And this matters because gene expression is fundamentally dictated by epigenetics and chromatin accessibility.
Let's explain chromatin accessibility, because every single cell in your body has the exact same DNA sequence, right?
Yes, your brain cells and your liver cells have the same DNA. But a specific genetic switch that regulates a liver enzyme might be tightly coiled around structural proteins called histones inside a brain cell.
So it's literally bundled up and physically blocked.
Exactly, making it completely inaccessible to transcription factors. It is permanently turned off in that specific tissue.
Therefore, a regulatory switch that drives a neurological disease might only be active in the deep tissues of the hippocampus. You will never find that eQTL in a TOPMed blood sample, no matter how many tens of thousands of people you sequence.
Because the chromatin in the blood cell has locked that specific regulatory region away.
So it's invisible.
Precisely. GTEx successfully captured tissue-specific disease signals that TOPMed was mathematically blind to due to its tissue constraints.
But on the flip side, GTEx lacked the massive sample size to overcome the statistical noise required to see the faint secondary switches and those crazy trans eQTL cascades in the blood and lungs that TOPMed illuminated so clearly.
Right. We require the deep dive microscope of TOPMed to unravel complex secondary and trans networks in accessible tissues. And we require the wide-angle lens of GTEx to capture the unique regulatory switches that only fire in highly specialized organs.
You can't just have one or the other. Solving complex genetic diseases requires integrating both the deep and the broad perspectives.
The synergy between deep transcriptomics and wide tissue sampling is really the only way we will fully annotate the non-coding genome. We are finally moving from a state of reading DNA like a dead language to actually watching the cellular machinery interpret the text in real time across the entire body.
That is an amazing way to frame it. So let's tie all of these threads together and summarize exactly what we have learned from this massive TOPMed dataset. The era of staring blindly at genetic dark matter is ending.
Truly.
By utilizing these massive sample sizes, researchers have proven that genes are governed by highly intricate control panels. The majority of complex disease risk isn't driven by obvious primary switches, but by tens of thousands of hidden secondary and tertiary regulatory pathways.
Furthermore, we established today that prioritizing global genetic diversity is not just an ethical goal, it's a strict mathematical and clinical necessity.
Right, uncovering over 11,000 novel regulatory switches in non-European ancestries highlights just how fundamentally flawed our older predictive models were.
Global health equity relies on diverse, transcriptomic datasets to ensure precision medicine algorithms actually work for the diverse patients who need them.
And finally, the discovery of those trans-eQTL cascades demonstrates that our genetic code is not just a simple list of independent instructions. It is an interconnected web where a single local change can send master regulator proteins across the nucleus to trigger massive biological responses.
Treating a complex disease often means tracing the downstream symptoms back to a single upstream master switch. We are finally reading the true physical wiring manual of the human genome.
Which leaves us with a pretty compelling final thought to consider for everyone listening today. We just spent this deep dive exploring how researchers uncovered tens of thousands of completely unknown secondary switches and complex cascades of genetic dominoes simply by increasing the sample size and diversity in easily accessible blood and lung tissue. So, imagine what unseen genetic dominoes are currently hiding in the deep, inaccessible tissues of the human brain or the heart, just waiting for a study with the immense statistical power of TOPMed to finally map them. The coordinates on the map are set. The real biological exploration is just beginning. The treasure chest was never empty. We just needed a much more sophisticated schematic to see the false bottom.
Well said.
Thank you so much for joining us on this deep dive into the hidden wiring of the human genome. We'll see you next time.